Welcome to Accelerate Science Now, where we sit down with innovators shaping policy, research, and industry to ask one big question: how do we make science move faster and work better for everyone? In today's episode, I'm joined by Jason Kelly, co-founder and CEO of Ginkgo Bioworks, a company that programs cells the way we program computers. We got into everything from how Jurassic Park inspired a generation of bioengineers to why programming DNA could reshape industries from medicine to agriculture, and how AI and automation might unlock a whole new era of discovery. Enjoy the show. Today's guest is Jason Kelly, a visionary at the cutting edge of synthetic biology and biotech policy. Jason is the co-founder and CEO of Ginkgo Bioworks, a synthetic biology powerhouse that treats cells like programmable pieces of hardware. Under his leadership, Ginkgo has helped develop new medicines, improve agriculture, create sustainable materials, and build tools to respond to global health threats. Jason started Ginkgo after earning his PhD in biological engineering from MIT, where he and his co-founders asked a big question: what if we could make biology easier to engineer? In addition to running Ginkgo, Jason is the former chair of the National Security Commission on Emerging Biotechnology, a bipartisan effort advising the U.S. government on how to stay ahead in biotech, especially as it overlaps with areas like AI and national security. Whether he's helping design vaccines, shaping science policy, or reimagining how we build with biology, Jason is focused on one thing: using biotech to solve real-world problems. So, Jason, thank you so much for joining us today.
Yeah, thanks for having me on.
Great. We like to really start out with, learning a bit about your background. I'd love to know if there's something specific, maybe a piece of technology, a class in school, whatever it was that sparked your interest and made you want to pursue a career in science and technology.
Oh, I know that very clearly, actually. I know because I went just this last weekend. There was at the Hollywood Bowl in LA. They did a Jurassic Park screening with like the whole concert orchestra, and it was amazing. And that movie came out like when I was 12 years old, or 12 or
Oh,
13
wow.
years old. And I
So
was like
that was it. That was the moment.
100%. Yeah, Michael Crichton fan through and through. I'd read like every book, and then I saw Jurassic Park, and I was completely sold. Yeah. So yeah, it's actually like among bioengineers in their mid 40s, it's like 80% are a consequence of Jurassic Park. Yeah.
Pop culture can have such a big impact on
It's huge. Yeah.
brains.
I really shouldn't be underestimated. I think it's a big pull. Yeah. Yeah.
Absolutely. And so, I mean, Jurassic Park, I'm sure most people, know the origins. I feel like it's actually kind of similar to what you're working on now. Ginkgo Bioworks is often described as the cell programming company. So
Yeah.
for people new to synthetic biology, maybe who didn't see Jurassic World or Jurassic Park, could you explain what you guys are doing at Ginkgo Bioworks and what that means?
Yeah, so I'll give you like the intro to bioengineering, right? So basic
Perfect.
idea is inside of every cell is digital code in the form of DNA, right? So it's ATCs and Gs, not zeros and ones like in a computer,
Mhm.
but you can read that DNA. So like you've heard of like the Human Genome Project, and that's a technology called DNA sequencing. So you basically like grind up a cell, put it into like a washing machine size device, a DNA sequencer, and then out comes on your screen the letters of the genome, right? So your genome would
Mhm.
be like about 4 billion letters long. A
Wow.
bacterial genome would be like a million letters long. All right. And so that's reading DNA. There's a parallel technology called DNA synthesis or DNA printing, where you do the exact opposite. You go on the computer, you type ATCGGG, you hit print, and out of machines at Ginkgo or companies out here in California like Twist Biosciences, a piece of DNA gets printed exactly to what you ordered, and then you can essentially, if you've heard of technology like CRISPR,
Mhm.
that allows you to find a spot in, say, that million letter genome of a bacteria, open the DNA at that spot you defined, and do sort of like a cut and paste. And you paste in the DNA you printed, and now you've essentially put in a new piece of code that makes that cell do something new.
Wow.
And that's the technology of synthetic biology or bioengineering. It's really that ability to change the DNA code in an organism, make it do something new. Uh,
That is so fascinating. And so you started Ginkgo Bioworks with a team of fellow MIT researchers over 15 years ago. Did
yeah.
you always believe that biology would become programmable like software, or is there a turning point where you started to think that that was something real?
Yeah, I mean, so the my experience was at MIT. So I was an undergrad at MIT, and I and did grad school there. But as an undergrad, I did all my summers like in the lab, trying to basically clone one gene into a cell. Cloning is the term of art for what I just told you, like putting a new gene into a
Right.
bacteria, and more or less failed. Like it's I was not great in the lab. Probably why I'm not allowed in the lab at Ginkgo. But it's unbelievably tedious. It's really hard to do. And so
Yeah.
I was pretty like demoralized by the end of like my undergrad. I was like, this doesn't work. And I met Professor Drew Endy, who I ended up working for in grad school, and then he was close with another fellow, Tom Knight, who I ended up starting Ginkgo with, along with three other students I went to school with. And their vision was what I just said. It was sort of, hey, we should really think of it like code, and we could program these things. And so honestly, it was just the fact that they believed it, sort of inspired our generation of students to kind of like go for it, and that became the field of synthetic biology. I will say, if you fast forward now, now I'm that was now like 20 years ago, 15, 20 years ago, and it's still hard, right? So, I think this is where a lot of, I think what your organization and what we're trying to do with like, accelerating science and some of the stuff that the National Security Commission on Emerging Biotech, that I chaired, we're working on was, well, what are the bottlenecks? Right. Like, because it is still like we don't see the pace in the biosciences like we see in computer science yet. And so what's holding that back? So I'd love to talk about that, because I think it is still not a solved problem. And
Yeah.
now, but the upside is if you solve it, right? Like, why is it so important to solve this problem, right? And I'll nerd out for 1 minute.
Please do. I think that's
Okay.
really valuable to understand where this could go.
Yeah, like why should people care, right? And
Yeah.
and so like what makes computers such a big deal, right? Like if you go back, rewind the clock 70 years, right? And computers were these sort of esoteric things, very small number of people were able to interact. Remember big mainframe, like that era of computing, like there was very few of them. And the reason that computers, even back then, you should have thought of them as a big deal was because they were programmable. So you take this device that you've invested all this effort in and you put in new code, and suddenly it does a new thing instantaneously. And I'll just point out, like, your car is not really like that, right? Like, you don't, like, insert new code into your car and suddenly the wheels change or something, right? Like, yeah, your gas system, or it has a computer in it, but the physical object is not really programmable, okay? But computers were programmable, so they could do all this different stuff by just changing the code, all right? However, it was limited, right? If you think about what computers can manipulate, okay, it is information. So what did computers disrupt? They disrupted media, telecom, finance, communication technologies, all the things that involve information, bits. What did they not disrupt? Hamburgers, right? Building materials, right? Like these are like the physical world, right? Okay, and that's because they don't move that stuff. Well, a cell, all right, is programmable. Like, I swear to God, like inside of a cell is DNA code. You can change it. You can make it do new things. You insert new DNA code and it can change color. It can make a new thing because it doesn't move information. A cell moves atoms. And the world we live in, you look around the room you're in, everything, it's all made out of atoms. It's made out of physical stuff. Cells are the only thing we have that are programmable in the physical world.
That's
All
so
right.
interesting. It makes
That's
me
why it
think of
matters.
it makes me
Like if
think of
we,
chameleons. Was that one of the things that you studied early on? I think I'm just thinking of an example of like something that can just change color.
like we
Like, are
just
they
peanut butter
changing?
food.
Yeah,
Yeah,
are they just
the
changing their programming? How does that work?
I mean, yeah, it's the like all the I mean, it's interesting you bring that up. So like one of the other thing that's amazing about biology is like there's the existence proof of
Yeah.
wild biology out in nature doing all these crazy things. Right. So again, I'll give you an example. Let's like let's over here. There's a plant in this window behind me. Okay.
Uh-huh.
Right. So let's imagine like we didn't have plants, and Tim Cook got up on stage at Apple and was like, hey, I've invented this thing, the seed. All right. And I'm going to tell you how this works. I dig a little hole in the dirt. I put this object in it. I cover it up with dirt. And then, with just the addition of air, water, and sunlight, this thing starts self-assembling physical atoms out of the CO2 that's in the air. It builds solar panels to collect more energy to do more of this. And then it starts to do basically nanoscale manufacturing, producing these little fruits at a level of atomic scale resolution that exceeds what TSMC or NVIDIA or Intel can do with semiconductors. It places atoms better than they do. All of that for free if you just put it in this hole in your backyard.
It
Right?
sounds insane when you put it that way.
It's literally insane, right? Like, like what is that technology? That's
Yeah.
crazy. So like that's the potential, right? The potential is, oh my gosh, that this is as a technologist looking at biology, so far exceeds what we can do with our human engineered systems. It's crazy. Right. And that's before you get to the fact that like we're made out of biology, our food, yeah, right. Like the atmosphere is produced all with biology. So there's just a level of capability. And we as humans, we kind of take it for granted because it's so familiar. Right. Whereas like when we see new like technology inventions in computer science, electrical engineering, and electronics and things, they all feel new and different. And so we kind of ignore this thing over here. But it is, in my opinion, the most important technology for humanity in the future is biological engineering, because it is the programmable technology that moves atoms. And we already know it can do freakish things. Right. We know it because we see it when we just look outside as a technologist and look at something like a plant. And so that's all the potential. But oh my gosh, there's so many cool things if you go around and look at what biological systems are already capable of. It's wild. So yes,
Yeah.
the chameleon is just one of them, but there are many things that biology does that's kind of incredible. So
Right. And you've touched on a few of these that, Ginkgo's platform is really focusing on so many different areas, from agriculture to medicine to materials.
yeah.
What is a project that you guys are working on that really captures what you're trying to do and why it matters?
Yeah. And again, the way to think about us is we're like the tool providers. So the
Hmm.
reason I can be in all these different areas that seem so disparate is that I'm providing you the tools to program and design biology to make it do new things. Okay. And so our customers end up going to market in all these different areas. But like a project we've worked on for years that's super interesting is providing the tooling and infrastructure in partnership with Bayer Crop Science, which is the biggest ag biotech company in the world. And they're engineering a microbe to produce fertilizer for corn and other row crops. And the idea here is, I don't know, you remember like crop rotation from like elementary school, like, oh, you plant this and then you plant the legumes or whatever, right? Like so the reason that they used to do that was that like crops like soybeans or other legumes, they actually have in their roots microbes that take nitrogen out of the air. Okay, the atmosphere has a lot of nitrogen in it, and make fertilizer for the plant. Okay. And so, and if you're like, I was my undergrad was in chemical engineering, like the pride of chemical engineering is the Haber-Bosch process. The Germans invented it. And it's basically pulling atmospheric gas in to a big chemical plant, burning natural gas, and then making ammonia, synthetic fertilizer, by the ton. Okay. And it's magical. And we dump all this fertilizer, and crops like corn, which don't have those microbes, get synthetic fertilizer and grow like crazy. And that's like modern agriculture, but it's not great. It's a lot of CO2 production. There's runoff. You have a lot of like local environmental issues. And so the question that Bayer posed in partnership with us was, could you engineer the microbes that live on the roots of corn to act like the microbes that live on the roots of soy? And and to your point about chameleons, like what's cool is we already know biology can do this. It can run Haber-Bosch effectively, like in the roots of soybeans. So why couldn't you also do it in the roots of corn? Right? Like
Got
we
it.
know it's got the capability, right? Does that make sense?
Yeah. So they come to Ginkgo with this question. Do you
Yeah.
then have scientists that are like studying the DNA code of the
Yeah.
soybean legumes and then are
Yeah.
trying to like, what's the next
Yeah. How
step
do we
there
do this?
after the problem?
Totally. Yeah.
Yeah.
So there's actually a pretty cool so Senator Todd Young from Indiana, he's now chairing the National Security Commission on Emerging Biotech that I chaired previously, and he just put out a series of like 4 bills based on recommendations from the NSCEB, and one of them is on this project to catalog the genomes in the national parks. Okay. And to answer your question, like what we do for this project is we do absolutely leverage the fact that people have previously sequenced the genome, read the DNA of the microbes that live on the roots of soybean. And that is what gives us the code that allows you to then go figure out how to program the microbes on the roots of corn. You want to basically like take the soybean microbe code and move it over. Okay. And like it doesn't work on the first shot. Like, so you need to try a lot of different things. It actually helps a lot to have seen other organisms that do this too. So that like soybean microbes aren't the only things that fix nitrogen, is what it's called. And so you want to have as many examples of that as possible. And so we really would benefit from bigger public databases cataloging all of the genomes out in nature that have all these incredible tools, right? Like the chemistry allowing you to just out just out in the wild grab nitrogen out of the air and turn it into like ammonia for the crop is wild chemistry. It's only because you have these proteins, which is kind of like the building blocks of cells, that are able to do that. And that was invented not by humans, that was invented by Mother Nature. And what we've done is just catalog that code, learn to use it, and then as bioengineers, we're repurposing it to something that could be, 5% of global greenhouse gas reduction and a great savings in local fertilizer runoff. And so but that's only because people have studied the biology, okay, in nature. And so it is there's a really powerful feedback loop that the government could enable, which is to study more of that biology in a high throughput way, generate these big public data sets so that people can then go design the next generation of biotech products using all that. And obviously AI and data science are a big part of that too. So anyway, but that's where it comes from. Yeah, absolutely. We go and study those things. Yeah.
Got it. Yeah, endless possibilities there. And you brought up AI. I'd love to hear how Ginkgo is utilizing AI to help with these, massive data sets and having to do multiple attempts at everything.
Yeah. Okay. So now let me get to what we've learned of 15 years of banging our faces into this because it's not easy. So, and I'll also bring up a little bit like what we're seeing, like across the biotech industry generally. So we obviously like are not an academic lab, we're industrial. We do industrial research in partnership with companies that are trying to commercialize biotech products. So that's the like that translational layer where people are kind of making new discoveries, but then they want to go commercial. That's the part that like that I know the most about. Okay.
Hmm.
And so, and I think we're in like a small, like, or maybe a decent sized crisis right now, I would say, across the biotech industry, that it's worth people tuning into this and, folks that are developing these policies in DC or otherwise like to understand. So, all right, let's look at biotech. So I just told you we want to program biology. It's amazing. It's computers in the 50s or 60s. I think it's going to be the biggest industry in the history of the planet eventually. All right. Okay. So like, why is it not today? All right. And so there's two problems. There's a technology problem and there's like an application problem. All right. So I'll get back to the technology problem. Let me just speak to the application problem first. So on the application side, genetic engineering got invented in 1978. There was a Nobel Prize. Herbert Boyer at UCSF moved the first gene from one species to another. Okay. And that was the birth of genetic engineering. And it was much older. It wasn't cool technology like CRISPR. There was no DNA printing. It was very old school, like cutting and pasting literally. But that was the beginning. And he founded a company called Genentech. And Genentech, the very first gene that got moved from one species to another that they commercialized was human insulin being moved into bacteria. And once you put it in bacteria, you put it in a big tank right down the road here in South San Francisco. I'm in San Francisco today. And you produced insulin. And type 1 diabetics, my dad's actually a type 1 diabetic, like who couldn't produce insulin since they were kids and were taking insulin from pig pancreases back then, could get human insulin because you took the human gene and you made it in bacteria and you grew it almost like you would brew beer, and out came human insulin. Okay. And so that was like the first application, like the first app on your phone, basically. The first app of genetic engineering was human insulin, and it was commercialized by Genentech and Eli Lilly. All right. I still remember like the bottles that my dad had.
That's fascinating. I never knew the origin of. Wow, that's
That's the
incredible.
beginning. That was the birth of biotech. Okay.
Yeah.
And so you had through the 80s, things like EPO, human growth hormone, a whole bunch of like other human proteins started to be made in bacteria. And then you got antibody drugs like for cancer and so on. And then in the ag space, you had companies like Monsanto and others pioneer putting genes in for things like protecting against certain pesticide, crop pests, and also for things like herbicide tolerance. Okay. And so those were like the big things. And then the last like small niche application was like in enzymes for laundry detergent. So, like cold water laundry detergent. Remember, we used to have to always run laundry hot. Anyway, cold water laundry detergent is because there are enzymes that break up the dirt, and they're made in the same way with a big engineered bacteria in a tank, and you add that to laundry detergent, you can save all this energy on hot water. Okay, so those are the three apps. And if you fast forward to today, overwhelmingly 90% of the applications for all of genetic engineering is in human therapeutics. Okay. And that's like okay. But human therapeutics take a long time to get to market. And they're just like they're still like they're a small industry compared to the totality of what you could do with biotech. Does that make sense? But I bring it up because they're kind of like the last line of defense for all genetic engineering. So when I gave you that vision earlier of like we could program cells and hey, you could grow a tree. Why don't you grow a house? Why don't you grow a new building material? Why don't we make semiconductor chips? Using biology. It's like why don't we have all this abundance of being able to grow anything we want through programmable biology? Well, that all is predicated on improving the tooling of genetic engineering. But in order to get money to invest in the tooling of genetic engineering, the apps have to work. You have to be able to make money on the end apps. Like you're not going to get Windows if you don't have Word. You're not going to get Apple iOS, okay, right? If you don't have like Uber and things. Like there has to be ways to gain value from the tooling if you're going to get investment in the tooling. Does that make sense?
Yes. Yeah.
So here's your problem right now. Like the US is basically in the process right now of losing the biotech, like the therapeutic drug app layer to China. Like it is like, so, and just so you know how this industry works, basically a biotech here in San Francisco or Cambridge, Massachusetts, those are the two like hotspots, has some smart scientists. They do some genetic engineering, they invent a new drug, and then typically what happens is one of the large biopharmas buys that drug. Okay. And they buy it like when they see a good result in a clinical trial. So they'll spend $2 million, they'll buy the drug, they'll take it to market, and you'll get, whatever, GLP-1 drugs or some new cancer drug or whatever. In the last 3 years ago, less than 5% of the drugs acquired that way were from Chinese biotech startups. First quarter of this year, 30%. Second quarter, 40%. Okay. So what's happening is the like actual technology, not for manufacturing. This is like the usual, oh, we're gonna like make low-cost manufacturing. No. This is the technology of discovery, the technology that really drives the tooling, and all the money that flows back to the companies doing that is starting to leave the United States. And so if we lose that, yeah, we'll lose drug discovery, which feels like a mistake to begin with. But what you're really gonna lose is genetic engineering. And it's very scary to lose genetic engineering
Can you
because
explain a bit more of like what would that mean for us to lose on that race of genetic engineering?
it would be like losing computers. Like it would be like as if the US hadn't led the information technology revolution. Like we hadn't had IBM and we hadn't had Microsoft and we hadn't had Apple and we hadn't had Google. Like the seed of that, which was really IBM, happened in China.
Yeah, I imagine that would have very different outcomes.
Yeah.
What
And
what can we
I would argue, again, because these are the computers of atoms, not the computers of stupid information, which is fine, but like who cares? Right? Like the world around us, like, oh my God, how many more TikToks can I look at? Right?
Yeah.
Like this, all the stuff that matters to us is made of atoms, right? Like and so like we can't lose like the atom the programmable atoms technology. That's such a mistake.
How do
And
we
so
turn this around? Do you have a suggestion?
I do. I
Okay.
have a
What
couple.
is it?
And this is, by the way, like the National Security Commission on Emerging Biotech. They have
Mhm.
there's a final report that came out maybe like 4 or 5 months ago. Some really good, a lot of good stuff written in there. I highly recommend people dig in on the report. But my like my short answer is the reason we're losing I think that biopharma thing is a great bellwether because it's very practical, it's translational, it's real money. People are buying these things. It's not like just some made up stuff. Like they are innovating and winning in China on with this technology. So how are they doing it? Okay, well, the advantage that they have right now is like the scientific talent, which is like the way we do this work today, like by and large, is you take a scientist, like someone with my training, PhD in bioengineering, who is then further trained in industry a little bit, then works at a lab bench with their two hands, pipetting. Okay. And what the word pipetting means is basically like you pick up a little thing. If you ever seen it on TV or if you've taken a lab class, it's like a very controllable straw. And you can like suck up liquid and squirt out liquid. And you use that to basically set up lab experiments. And then you engage with a series of like benchtop equipment to like move this stuff around. You're kind of like a fine chef. Okay. And you know these like really careful protocols. It's all very precise. Remember, I told you I was terrible. Like this is stuff I'm horrible at, right? Like I'm ADHD, like I can't like, like the details. I don't get
It
the
sounds
details.
hard. Yeah.
No, it is hard. Okay. It is hard. And it's very much like a skill-based thing that you need to like learn from mentors and be trained on doing the science, and it's by hand. All right. So here's what happened over the last 20 years. First off, we train a lot of Chinese scientists. That's great. Actually, I think we should be like we should be pumping in smart people in this country. We're super good at it. A lot of them stay. But then China had a program very deliberately to bring people back. Okay. Number one. Number two, China's built up their whole academic infrastructure. So they have great schools now. And so, like, that scientific talent base, there's no longer this advantage we used to have where we just had more scientists. Like, China has more people, right? And they have more scientists. There's like just more physical, like human being scientists with great training. They're very smart. And so, like, that whole, like, that advantage is now gone. So if your strategy, oh, and by the way, like, they're working at it's maybe 40% cheaper. Okay, right? So you're getting like twice as much research per dollar out of just as talented people. And I don't see how we dig out of that hole. Okay, like producing more scientists doesn't really do it. Like like we got paying everybody less doesn't really do it. Rents aren't going to be less in Cambridge than they are in wherever in China. Like none of that's going to do it. So I, like the way out, in my opinion, it's kind of like how I think the strategy we have to adopt to bring manufacturing back is automation. So like what we need to do, like the US bet needs to be a bet on laboratory automation. All right. And this is going to tie into the AI stuff. But like if we think we're going to beat China with hands in the lab, like I got a message for you. Like it's not going to happen. Like there's just as many there are more smart scientists, just as talented in the lab, working harder for less money. Like it's not going to happen.
Yeah. How do you compete with that?
How you can be right? Like let's just look at the numbers, right? So our road out of that is what I'll call total automation of labs. In other words, you need the lab to feel like basically a warehouse with all that same laboratory equipment that you were interacting with as a scientist at the bench is now all connected through integrated robotics. And you can, with a software layer, send a sample to any piece of equipment in that big setup and have it go execute whatever it was going to do and then get sent to the next piece of equipment without humans in the loop. Okay. And that, if you might have seen in the America's AI Action Plan that came out of the White House just a couple of weeks ago, which is also excellent. They have a whole section on accelerating science, and they call these cloud enabled labs. Okay. And that's what they're talking about with it's basically, it's really, you should think of it like a doing the laboratory work without the bench scientist.
Yeah.
And
No more scientists pipetting.
no, scientists can think. Let me tell you what. Let me tell you what. Like So, I think this is actually a really important point. Like the you have. I'll give you a different analogy just to put a point on it. Back in the 1970s, the computer science department at MIT was not the computer science department. It was the electrical engineering and computer science department. And it was one group. Because in order to be a computer scientist, you also had to be an electrical engineer. right? Because like, how the hell were you going to program a computer if you didn't like know how the whole thing worked and everything, get in there with the wires and all this crazy stuff? And what we did in the interim period of time was we split electrical engineering and computer science into two different things. And we let the electrical engineering people go invent semiconductors and they're all about, automation and scaling stuff up. And the computer scientists, instead of having to have their hands dirty all the time with the wires, just got to think all day.
Yeah,
Okay?
that's a big change.
And it's extraordinarily enabling because it turns out the culture of messing with the wires and what ultimately became like lithography and, planar semiconductor manufacturing and TSMC is a super different culture than the computer scientists that became object-oriented programming and operating systems and now neural nets and, all the AI stuff. Like they're very different culturally. Okay. Right. Like it's all math nerds and this is all like physics nerds. Right. And right now in genetic engineering, if you want to design DNA, if you want to be the world's best biologist and understand the ins and outs of that nitrogen fixing bacteria and be really smart about the DNA, boy, first thing you better know how to do is pick up a pipette and move clear liquids around very carefully at a lab bench. It's
I don't know.
absurd.
Are you worried though
You
that
got to split
scientists
them.
will lose this trauma bonding of pipetting?
That is actually not a bad point. There is real trauma bonding in the lab. My God, you really feel like you earned it. But the dream is, and by the way, like electrical engineers were like a little bit bothered by this too at the beginning when the computer science really got split off. Because it was like, oh, they don't really know what's going on. Like as you abstracted the two away, like there was, but like, oh my gosh, was that enabling? And so I think we scientists should be excited to get that split apart. And anyway, certain scientists freaking love thinking about how to make the lab, like the lab work more standardized, more scalable, more automatable. Like there's a whole culture, just like there are a culture of electrical engineers, of a certain type of scientist who gets excited about the physics of improving the throughput and scalability of lab work with automation. But then there's a different type of scientist who's just like, all I want to think about is the DNA, the biology. I wish I didn't never had to think about any of that stuff. Just give me the output. Right. And so my belief is like the way the US gets out of this like puzzle around genetic engineering and losing it to China is we got to split those things. And then we got to really be the experts in building the automation. And then also because that lets you move faster, now we get to be the people who develop kind of the moral equivalence of the programming languages and the operating systems for programming DNA. And that's all like right up our alley. Like we could lead in both of those things, no problem, because they're not a function of how many talented hands you have. Like we're
Right.
not going to compete at talented hands. Like that's like not the US's strong suit.
No.
Like low cost talented hands. Like what the fuck?
Yeah,
Right?
yeah.
That's the China thing through and through. So I do think that's to me the biggest thing. And so like at Ginkgo, we've been doing a lot on that automation side. We have like the world's we actually just did a deal with a national lab, PNNL, Pacific
Mhm.
Northwest national lab, to build a like 18 instruments inside an anaerobic chamber, all controlled by robotics. And the reason that needs robotics is like people can't be in the anaerobic chamber. There's no air. Okay. And so you connect 18 pieces of equipment all together with we have like a cool system that like, it's just like like little tracks that deliver the samples and robotic arms in front of every piece of equipment. It's really neat. But like you kind of are at this point, like where you see automation is like when you have to have it like in an anaerobic chamber. Okay. And my view is what we really should have automation is in every academic institute, in every biotech company, in every company, like everywhere you're doing lab work should be replace the benches with cloud enabled labs with these like big automated facilities. And that gets us out of this. In
Right.
fact, you have to do it. Like if we don't do it, we're going to lose.
And so, understandably, a lot of people are scared about AI replacing jobs. And I think you're talking about, you're framing it in a really helpful way that it's not that it's replacing jobs, it's that it's making them different. It's that, like, you can do the things that you're actually really good at versus pipetting. Is
100%.
that true for the whole lab, or are there any instances where you think that that's different?
No, I think that's, to me, that is like, my view on it is splitting these things apart. There'll be more that like the people that love lab stuff can get into this half of the house, right? Like there will be like a lot of work to do on like actually like today you can't you can't make automation do everything you can do at the lab bench today. That there's a 5 or 10 year technical journey. You can do more and more every year, and you should move more and more of the work there. And like that's all going to be critical and extremely valuable. But there's like a ton of jobs for people that are excited about that. And then I think on the other side, there's a million scientists who their whole career path is to get out of the lab, basically today. And you should just be able to move that back to earlier in the career. Right? Like right now, your goal is to manage a team of 15 people who all have to work with their hands, but you don't have to work with your hands anymore. It's like, no, no, no. Like you should be able to come right out of your PhD, learn all this stuff about biology, and primarily be working on designing DNA and ordering your experiments at the computer. Like that I think no one should be too upset about that, in my opinion.
That's fair.
Yeah.
So you've talked about a few things that the U.S. government is doing or proposing, from the AI Action Plan and cloud-enabled labs to Senator Todd Young, right, who has
Yeah.
the new data sets from
Yeah. And he's also doing
national.
the cloud lab bill too coming, I think,
Oh,
also.
really? Okay, perfect. Well, yeah, I was going to ask, besides, it makes complete sense that these cloud labs and having huge data sets are really important for moving forward in biomedical engineering and bioengineering. Is there anything else that you think is missing from that conversation that we need to focus on?
Yeah, so I think the third leg of that stool, so it is like, so there's like, I want the lab to the lab work to be able to get cheaper with scale, and the US to be able to do more of it than China. The only way out of that puzzle is automation. I want more data to be smarter about biology, to train AI models, okay, that speak DNA. Then I need more data sets like the National Parks Genome Project that Senator Young is proposing. The third leg of the stool is, if you have the first two, particularly if you have the first one with the lab automation, is can you use the reasoning models? Okay, so like the GPT-5s, like whatever, that things that can think, Gemini, and have them control the lab also. Like, could they design experiments? Could the models design experiments? And then basically get data back from the lab that serves as like reinforcement learning to the model so that it gets smarter based on what it's learning. And I get really, so I think this is a way of thinking, like those reasoning models plus an automated lab is a way of thinking about not just bioengineering and genetic engineering, but all of science. And the reason I think this is worth thinking about is, a lot of what science is doing is scientists are doing is sort of like becoming experts in a very narrow domain, and then kind of running a series of pretty logical experiments to drill down into that domain and to ask a question that like no one's yet asked in that domain. And mostly the questions we ask are the ones that scientists with good taste who know a lot about that field think are like the most important next questions. And they're very limited in how many things they try because they've only got two hands. I think there's a slightly different model where you maybe have a scientist sort of like directing, like, hey, these are the parts of science we want to push on next that are most important to humanity, or blah blah blah. And then you set, like, they have like sort of like agentic scientists working for them who are gonna like go and push down an experimental line, inclusive of doing the lab work, and come back after a few weeks of doing experiments and say, here's what I learned along that question you wanted me to ask. And I think a lot of that work is doable by reasoning models, because
Hmm.
it's not the like really high end science stuff is understanding the limits of knowledge and where to ask the next question with good taste
Right.
and so on.
What questions
And
to ask?
rather
And
than chase it up, right? Like with a lot of workaday experimental work and kind of reasonable conclusions based on what you see from the data. And I think a lot of that is like you're kind of using like big brains to do like not the most brainy stuff when you're
hmm
forcing science to go through that loop.
hmm
It is, but sometimes it's not. And so I think there's going to be a certain class of problem where we could just blow it out. Like we could have, a single scientist running 5,000 agentic scientists with a research budget of reagents running through an automated lab, and like, my God, what could they figure out, right? So there's really cool stuff like that is well, I mean, right now people are just trying this stuff for the first time. Like, we'll see. But if that's a great way to think about just kind of a totally different way of doing science in the first place that I think could be enabled. And it's not requiring a miracle, right? It's not requiring like a super genius, um, reasoning model, because I'm not telling you go look at everything, read all the literature, understand all human knowledge, and then come up with something that no humans figured out. I think there'll be some stuff like that, but like humans have also looked at all that stuff, and we're pretty smart, right? I'm letting you I'm letting you do what scientists do, because this is kind of how we cheat to discover new stuff. We don't think big thoughts all the time like Einstein did. There's like the pure theorists, but mostly what we do is experiments. Like, we go out and actually just do something for the first time in the world, and what I share with you, all this my brilliant science, is just the result of trying it.
Right. And so if
So
you have someone to help try way more
if the
things, you
model
can.
can try things with an automated lab, why can't it discover something new? Most of discovery is trying new things, right?
Right.
Like, and so it's actually that's to me the block more than the thinking big thoughts is, can you give it hands in the lab? If you give it hands in the lab, it's going to discover stuff left and right, because like most things in the world haven't been tried, right? Like, and so like so there's a whole thing there that I think is like, wow. At that point, you're just sort of like you're kind of thinking of your budget more about the consumable costs and the cost of like running the equipment, like equipment time plus consumables time versus like big brain time, right? Which has been kind of the limiter on science to date. It's mostly been think, 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 do one little experiment, because like the experimental time is so precious because it's so painful. Does that make sense?
Yeah. Oh, it makes complete sense and exactly why that could be so helpful. I think it might, as we wrap up here, it would be helpful for you to illustrate. So if we get this right, combining biology and AI, what do you think becomes possible in the next 5 to 10 years that people might not be imagining yet?
Yeah. So I mean, I think there's like I'll give you what I think is the near end stuff that people can get. Then there's like the long term. So like the near end stuff, I think there's a huge amount of stuff in health, and I think there's like two great projects. There is finish up disease. Okay, right? Like, we have been on a journey, like, I don't know, my kid's got a skin infection that's going around this summer, right? It's like, you gotta scrape on the turf, and then he got like a fungal infection around it. And it was like, okay, wasn't going away, wasn't going away, wasn't going away. And you go to the urgent care clinic and they're like, oh yeah, this is an infection. Here's the oral antibiotic he needs to take. And it costs nothing. It's generic. It's been on the market for 50 years. It's like a $5 thing. And like, and it just goes away. And that kind of like infectious disease of bacterial, like those are things that used to kill kids, okay, 80 years ago. So that's one that we don't even think about today, but we just like, we crossed it off. Okay. And there's still other big ones left, cancer we haven't crossed off, Alzheimer's we haven't crossed off. But like, there's just a list. And one big human project is cross them all off. Okay. So that's one on the disease side. And boy, let me tell you, bioengineering is the biggest hammer in that toolbox by a freaking mile. Okay. So that's one. All right. And then the other, I think the GLP-1 drugs are like the harbinger of this, right? Because like, hey, GLP-1 drugs, oh, it's obesity. It's like, it's not. They added $1 trillion of market cap, not to treat obesity. I'm on GLP-1s. Like, it's a weight loss and longevity drug. It's awesome. Like, you don't think about food. It's great. It's like mental health, awesome, and it's like all course mortality is improving across the board, right? And so like this thing is the first longevity drug, really, of like great consumer awareness, and like that is the next big project. It's like, well, that goes on forever. Okay, right? Like, we can keep, more stuff is going to come out. How long do you want to have muscle mass into your 50s, 60s, 70s, 80s, 90s, 100? Right? That's so, and like all that is untapped, untapped. We don't even have the regulatory right now. Like, everything has to go to market backdoor. It's like, oh, we're treating this, da 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 da, da. And then like, oh, by the way, it also might just be useful to like, I don't know, 500 million people or something who are above the age of 40, right? Like, so there's sort of like a big opportunity. So those are the two near ones. And then in the long run, it's all the atoms. It's everything. Who knows? It's like, it's the stuff your kids are going to be excited to do, right? Like, like it's Jurassic Park.
Right. I
It's who
that
freaking
was
knows?
maybe
It's
my last
computers in the
question.
yeah, I
If
grew up when computers were becoming personal, right? And like, so I got to think about all of the cool things you could do with computers as a kid. Like, if we do our job right by improving the tooling for genetic engineering and design biology, our kids will get to imagine all the things. And I don't know what they'll come up with, but it'll be awesome. So,
if a company came to you knowing what you know from reading the books and watching the movies and they wanted to create Jurassic Park, would you help them?
oh, 100%. Yeah. 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 there's a company called Colossal that's working on it. Yeah. Oh, definitely. Yeah, of course. Everyone. The reason Jurassic Park is such a great book, by the way, is it like teaches is that there's two important lessons in there. One, is like life finds a way, right? Like we didn't design biology, so you better respect it. Okay, like watch the hubris, scientists and engineers, people like me, right? Like watch the hubris. So it gets that across. But then importantly, like that first scene, right, when they pull up on the Jeep and they look out and there's like the brontosaurus, like it's the majesty, right? Like biology is majestic. It's powerful. We're made out of it. It's the two together that is the future. That's the right way to enter the bioengineered future. It's like respect, like, oh my gosh, we have to get into it. Look at the majesty, but do it with humility.
Mm-hmm. Wow. Well, I can't
Yeah.
think of a better way to wrap up this conversation than with that. So thank you so much for joining us today, Jason. This has been incredibly illuminating.
Thanks, Adeline. Yeah, enjoyed being on. Appreciate the
Great.
time.
Thank you. You've been listening to Accelerate Science Now. I'm your host, Adeline DeYoung. This show is produced by Kindred Subjects, Accelerate Science Now, and Seed AI. Our executive producers are Josh New and Austin Carson. Special thanks to Kevin King for editing and production. Thanks for joining us, and we'll see you next time.